Hello there. Today in this lecture talk, let us understand question and answers related to nucleic acids. So what are nucleic acids? Nucleic acids are biopolymers or large molecules essential for all known forms of life. Nucleic acids, which include DNA, that is deoxyribonucleic acid, and RNA ribonucleic acid, which are made from monomers known as nucleotides. Okay. Then what are nucleotides? Nucleotides are composed of nitrogenous base, a 5-carbon sugar, ribose or deoxyribose, and at least one phosphate group. So if you get any question like what are nucleic acids, you can write this answer. I hope you're able to understand very clearly. So what are nucleic acids here? Nucleic acids are biopolymers, are large molecules essential for all known forms of life. All right, please make a note of this about nucleic acids. Then coming to further in detail, can also define these nucleic acids as the biopolymers that are the hereditary dominants of living cells are known as nucleic acids. Okay, the nucleic acids are the vital components of living system. They are responsible for storage and transmission of genetic information, right? And they are found in the cells. So we are able to differentiate the nucleic acids, that is DNA and RNA, based on the nature of the sugar present in them. There are two types of nucleic acids. Those are deoxyribonucleic acid and ribonucleic acid. That is DNA and RNA. I hope I made very simple this particular nucleic acid. Then if anybody asks the question, what are the components of nucleic acids? Okay, so there are basically these nucleic acids which are made up of 3 components. Okay, the first component, pentose sugar, nitrogenous bases, phosphoric acid, H3PO4. The first one, pentose sugar. In DNA, that is deoxyribonucleic acid, the sugar is 2' beta deoxyribose, and in RNA, beta D ribose. Okay, this is the first component of the nucleic acid, that is ribose, where at the 2' carbon atom, hydroxyl group will be there. Whereas in 2' deoxyribose, at the 2' carbon atom, there is a divide of oxygen molecule. That's why this riboses are called deoxy. There is a removal of the oxygen group. This type of sugar moieties or sugar component can be observed in the DNA molecule. So it's deoxyribose nucleic acid. Okay. Whereas here in the ribose, at this is the first carbon atom, 2nd carbon atom, 3rd carbon atom, 4th and the 5th carbon atom. In ribose, at the 2nd carbon atom, there is a presence of hydroxyl group. Whereas in DNA molecule, in the sugar molecule, at the 2nd carbon atom, absence of oxygen molecule. So it is called deoxyribose. Why this 2 mentioned here? 2 because at the 2nd carbon atom, there is a deoxy. Oxygen molecule is removal, that is deoxy. That's why this is mentioned as 2' deoxyribose. All right. So this is about the sugar, that is pentose sugar. Okay. 5 carbon atom, that's why it is called pentose. The next, coming to the nitrogenous bases. Second component of the nucleic acid is nitrogenous bases. So these are the nitrogen containing heterocyclic compounds with slightly basic properties. Okay. So as the name suggests, nitrogenous bases mean there we can observe the other than carbon atom, nitrogen atoms will be there. Okay. Why we called here heterocyclic? Because other than carbon atom, there is the presence of the other common other compounds. Okay, so that's why it is called heterocyclic. Not only carbon, there is a other than carbon, nitrogen is also there. That's why heterocyclic. Other compounds are there. And what are these nitrogen bases? Basically, these are the derivatives of the 2 parent heterocyclic compounds, purines and pyrimidines. Okay, in the exam, you may get what are nitrogenous bases, or maybe what are purines, or maybe a pyrimidines. So now let us understand what are these pyrimidines. So pyrimidine bases are nitrogen-containing heterocyclic compounds with six-membered ring, and they are derivatives of pyrimidine. Okay. So what are the pyrimidines? Examples: cytosine, thymine, uracil. Uracil in RNA molecule. Okay. Instead of thymine, there will be the uracil molecule. So all these three which comes under pyrimidines. So whatever the information is here, that can be written for your pyrimidine and also for the purines. So if anybody asks what are purine bases, so these purine bases are nitrogen containing heterocyclic compounds with the 9-membered double ring, whereas pyrimidines are single ring, that is single ring with the 6-membered ring. Okay. Whereas nitrogen containing heterocyclic compounds with the 9-membered double ring is purine bases. The two purine bases that are found in both DNA and RNA are adenine and guanine. Okay. This adenine and guanine can be observed in both DNA and RNA, whereas here only uracil is in the RNA molecule. Cytosine and thymine are in the DNA molecule. All right. I hope I made myself clear about pentose sugar, nitrogenous bases, pyrimidine, and purines. Now let us go through the structural representation of purines and pyrimidines. As I told here, this particular pyrimidines, which is a 6-membered single ring, right? This is the 6-membered. 1, 1, 2, 3, 4, 5, 6. All right. So it's a six-membered single ring. Okay. So this the 6-member are there. Other than carbon atom, there's a presence of nitrogen. So we can say this, both purines and pyrimidines are heterocyclic. For pyrimidine example, cytosine, uracil, and the thymine. All right. So they are the derivatives of the pyrimidine. So these are the three nitrogenous bases, nitrogen containing heterocyclic compound. Hetero because other than carbon atom, nitrogen is also there. Then coming to the purines. What we told about the purine? These are nitrogen containing heterocyclic compounds with 9-membered double ring. 9-membered double ring. So this is 1, 2, 3, 4, 5, 6, 7, 8, 9. 9-membered heterocyclic, because other than carbon atom, there is a presence of nitrogen. So it's a heterocyclic 9-membered double ring. This is the first ring and this is the second ring. So example, adenine. So this is the adenine and this is the guanine. So this is the parent structure of purines and pyrimidines. Okay, this is the parent structure of the purines and pyrimidine. And also we can represent this one in a IUPAC names of purines and pyrimidines also observed. So for this particular purine, in purine, the adenine can be mentioned as 6-amino purine. Okay, why we mentioned a 6-amino purine? Because this is 1, 2, 3, 4, 5, and 6. Of the 6th carbon atom, there is the presence of the amino group. So you can mention this as 6-amino purine. You can keep in your mind parent structure. At the 6th carbon atom, add the amino group. Then that will become adenine. Next one is the derivatives of the purine is guanine. So how we can mention this guanine as 2-amino 6-oxypurine. 2-amino because this is 1, 2, 3, 4, 5, and 6. This is 7, 8, and the 9. So additional extra component can be observed, compound can be observed at the second carbon atom. Second carbon atom, there is the presence of the amino group. So 2-amino. At the 6th carbon atom, there is the presence of oxygen molecule. So 2-amino 6-oxypurine. All right. Then coming to the pyrimidine. So in this particular pyrimidine, we could see here the cytosine. So cytosine can be mentioned as here, this is 1, 2, 3, 4, 5, 6. So this is 1, 2. At the second carbon atom, there is an oxygen molecule. So 2-oxy, 4-amino pyrimidine. Okay. 2-oxy, 4-amino pyrimidine. At the second carbon atom, oxygen molecule. So 2-oxy. Then at the 4th carbon atom, amino group. So 2-oxy, 4-amino pyrimidine. Next one is uracil. How we can mention this uracil in IUPAC name? That is, this is 1, this is 2, 2-oxy, this is 3, and this is 4. So oxygen molecules at the 2 position, you can say 2, 4 oxy. Okay, not oxy. There are 2 are there. So dioxy. 2, 4 dioxypyrimidine. Okay. The next one and the last one is thymine. Okay. The thymine, this is the first carbon atom, second, third, fourth, fifth, and the sixth, 6-membered ring. So how we can mention here 2, 4 dioxy. 2, 4 dioxy. Apart from that, at the 5th position, there is a methyl group. That's why we can mention 2, 4 dioxy 5-methyl pyrimidine is thymine. All right. So this is all about the structure representation of the nitrogen containing nitrogen bases of the nucleic acids, that is in purine, adenine, and guanine, and whereas in pyrimidine, cytosine, uracil, and the thymine. Okay. So far, what we went through in the beginning about this particular, what are basically nucleic acids, right? Or the nucleic acids and the components of the nucleic acids, pentose sugar, nitrogenous bases. Okay, we went through. Then the next one is phosphoric acid. As the name suggests, phosphoric acid, there's the presence of one phosphate molecule, right? One phosphate molecule, right? One phosphate molecule and 3 hydrogen. H3PO4. 3H, then 1 phosphate molecule, 4 oxygen molecule. So, phosphate molecule, that is H3PO4, which is a part of nucleic acids. Okay, and also part of components of the nucleic acid. So, what are the components of nucleic acids? Pentose sugar, nitrogenous base, phosphoric acid. This phosphoric acid play a very important role in the formation of phosphodiester bond. Okay, this phosphate molecule will attach at the 5th carbon atom of the ribose sugar. That's why we say 5' phosphate. Okay, at the 5th carbon atom, there will be attachment of the phosphate molecule of the ribose sugar, or maybe a deoxyribose sugar. So this is the ribose sugar. At the at the 5th carbon atom, there will be an attachment of the phosphate molecule. Okay, and 5th carbon atom, and at the 3rd carbon atom, there will be again phosphodiester bond formation. Only these 2 regions are there for the formation of the phosphodiester bond. So these are the carbon atom number, what we say as 5' phosphate molecule, 3' hydroxyl molecule, like that. Okay, 5' and the 3' molecule. Okay, so this is all about the nucleic acids and about the components of nucleic acids. I hope I made myself clear about this particular nucleic acids, components of nucleic acids, purines, pyrimidines, then ribose sugar, and the phosphoric acid molecule. Thank you everyone for watching this video. Stay tuned with me.